chapter 3, talking about cells is really such a foundational part of everything we're going to talk about. If we don't understand what's going on at the cellular level, then it's really impossible or very hard to picture what's going on at every level above that. So we need to have a really, really basic but thorough understanding of what's going on at the cellular level, because most physiology is happening at the cellular and subcellular level. It's going to affect every other level after that. So we're going to spend a little bit of time on chapter 3 to make sure you understand some of the basic principles that's going on inside the cell. Chapter 3 deals with cells and tissues. We'll mostly be covering cells in lecture, and we'll talk more about tissues in lab. And I think we've already talked about tissues in lab, but we'll tie them together. But the most important thing about cells is it's really the lowest living level of organization, or in other words, it's the lowest level of organization that's considered to be living. If you go smaller, such as the organic or inorganic molecules, such as proteins or water, they're not considered to be living in their own, but they certainly contribute to the conditions that would be necessary for living in the actual cell. So most of the metabolism, most of the chemical activities that we talk about are occurring at a cellular level. And of course, we know cells are made up of organic and inorganic molecules, and cells make up tissues. Cells, of course, as we say, are the lowest living thing, but not every cell is exactly the same. There are many different types of cells in the body, and we'll be looking at those as we get into the different systems that they belong to. But right now we want to talk about what is it that all cells have in common. And so if we understand this theoretical construct that we call the general cell and the parts and what they all have in common, then when we talk about the actual cells, we can talk about the differences. So what all cells have in common is three main things. They are surrounded by a cell membrane, also called a plasma membrane, that controls what can go inside and outside the cell. It actually separates one cell from another, or the cell from the material outside of the cell. It's made up mostly of phospholipids. It really has a great deal to do with what physiology is all about, so we'll talk more about that. Then inside the cell we have the nucleus, and its primary mission is it stores the DNA, the instructions for building our proteins, but there are other parts of the nucleoplasm, and we will talk about that. And then there's the contents of the cell inside the membrane but outside the nucleus, and that's the cytoplasm. The cytoplasm is basically made up of two major structures: cytosol, mostly water, and other dissolved things in the water, and then the organelles, or the structures within the cell that perform different functions. Okay, looking at the nucleus, and of course the nucleus is where the DNA is kept, but not just DNA, also even individual nucleotides of both DNA and RNA, and there are proteins in there. But of course the DNA is really important. It wraps up from being unwound as chromatin into winding up to be chromosomes before a cell can divide. So our genetic information is found within our DNA, and we need to express certain genes to build certain polypeptides and proteins within the cell. The nucleus is actually surrounded by a double phospholipid bilayer called the nuclear membrane, and it actually has pores called nuclear pores, so things can actually go into the nucleus or things can go out of the nucleus, as we will see, very important to what can go on in a cell. We also have the nucleolus, which is important mainly from the standpoint that it's how we produce our ribosomes, and of course ribosomes are the factories where we build our proteins. So without the nucleolus, we don't have ribosomes. Without the ribosomes, we don't have proteins, so it has an extremely important function. Then again, the majority of the DNA is in the form of chromatin, unwound DNA in the nucleus. As we said, the nucleoli, there can be more than one, but its main job is that's where our ribosomes are produced, and then they can actually move through the nuclear pores and either go and attach themselves to an endoplasmic reticulum, or they can even float around freely in the cytoplasm. Again, the chromatin is just basically the unwound DNA and protein, and it's all found all throughout. And of course, most people have heard of chromosomes, but what they may not realize is that the chromatin is the DNA, and it actually winds up using proteins and some other chemicals, winds up to actually form chromosomes so that the DNA can actually be divided into two cells, and both cells can have the same amount of chromosomes and DNA. The plasma membrane really has such an important job of controlling what can go into and out of the cell, separating one cell from another, just forming this semi-permeable barrier, and that it has what's called a phospholipid bilayer, which I like better than the term double phospholipid layer. The bilayer tells me that I have phospholipids that the heads face outward and the tails face inward. Because the heads are charged and polar, they're attracted to water, which is also polar. But the tails are not charged, they're fatty acids, so they want to be away from water, so they turn inward. But even though by molecule the phospholipids are the most numerous molecule, really the plasma membrane also contains some other extremely important structures. We'll talk about what they do. By weight, proteins would actually be the largest structure in the phospholipid bilayer membrane of the plasma membrane. But we also have structures like cholesterol and glycoproteins, which also have important functions. As we see in this picture, if you notice, the most numerous molecules in this picture are the phospholipids. Notice how the heads are pointing outward to the extracellular fluid, the interstitial fluid, and notice how the heads are also pointing inward to the cytosol, the fluid within the cell. And notice how the tails are pointing inward, forming this barrier. Now, also though, notice that there are these purple structures. There are many different proteins that do many different jobs. We'll be discussing some of those jobs. A couple of the jobs we can see in this very picture, we can actually see some of the proteins that look like they're serving as passageways, doorways, channels that are allowing certain things to either enter or leave the cell. And we see that there are some proteins that look like they're actually anchoring proteins that are holding the phospholipid membrane to the actual cytoskeleton. And then we actually see our next group of molecules, our glycocalyx, which include the glycoproteins, the proteins, the purple structures with the sugar groups hanging off of them, and the glycolipids, also the fatty acid tails with the sugar groups hanging off of them. And they're really very important. They're really how our body identifies our own cells. Just like we all have different fingerprint types, we also have different glycocalyx within our cell groups, and our body learns to recognize what our glycocalyx look like on our cells. So if something comes into our body that is not from us and has a different sugar group marker, our body will usually reject it. We also see cholesterol that's embedded in that membrane. And of course, cholesterol, a lot of people give it a bad name, but really cholesterol is a needed molecule, and I like to compare it to, is air in your tire a good thing or a bad thing? Well, the truth is, you need air in your tire. Too much can be a bad thing, too little can be a bad thing, so there's a correct amount. Same thing with cholesterol. Here we see something that we have often in cells, especially when we need to increase their surface area to either absorb something or secrete something, usually absorption though. And of course what that is are the microvilli, or the folds at the top. So anytime we have a small amount of space but we need a lot of surface area, how we solve that is we fold the surface area. So in these epithelial cells we see here, they are probably being used to absorb something, and they need a large amount of surface area to absorb the amount that actually would keep the cell alive or do the job of whatever the cell needs to do. It will need to absorb certain things to do its job, so there are microvilli that allow us to do that. What we also see in this picture, in these epithelial cells, because we see a free end on one side, we see a basement membrane connecting them to usually some sort of connective tissue, and we see that the cells are anchored together by different types of connections. And those different types, basically what we see in this picture is we see some tight junctions, which is almost like protein glue that's holding the membranes very close together. We see some desmosomes, which are basically like anchors. We see some connexons and some gap junctions, which basically are like little tunnels that, in the gap junctions in particular, will allow things to, such as ions, to pass from one cell to another. And that's important, especially in cells like cardiac muscle cells. Okay, so our cytoplasm, again, is really broken down basically into two major things, and they include the inclusions, which I haven't really seen that much in most literature. But anyway, the two things that we know that's in there is the cytosol. We know that 62% of our water in our body, approximately 62%, is inside our cells. So it would be the main part of the cytosol would be the water, even though it's not pure cytosol. There would also be the other solutes and other things that are in the cytosol, ions or even non-charged particles that may be in the water. We also have the organelles, which is basically the machinery of the cell. The literally means little organ, the parts of the cell that have specific functions for the cell to function properly. Here we actually see a cell, and need to be familiar with this picture, even though we need to be familiar with the structures and be able to apply them to different pictures or even different microscope slides. This is the picture from your book, so it's the picture we will be using in this class, but understand there are other representations, but they all have certain things in common. So one thing that we see here, we have our three main structures or membranes surrounding the outside, even though we've cut through the membrane so we can see what's inside. We see our nucleus. We can see the inside of the nucleus, see the chromatin and the nucleolus, and all those structures together make up the nucleoplasm. We can see the cytoplasm inside, and of course the cytoplasm is going to contain the organelles and the cytosol, and we can see these different structures such as endoplasmic reticulum, both the rough because it has ribosomes, the smooth because it doesn't. We know that proteins are made of ribosomes, so we know that they have something to do with making proteins. We can see our Golgi apparatus, which is kind of like a final processing and packaging plant full of enzymes to finish structuring chemically things that we're building in the cell. And then those things can either be used for the cell or they may be exported to somewhere else through exocytosis and secretions. We also see centrioles, which are necessary for a cell to be able to divide. See our mitochondrion, which does one of the major functions for most cells, and that would be to make a great deal of ATP. And of course, the reason why mitochondria can make so much ATP is because they have so many proteins called ATP synthase enzymes inside of them. So you're responsible to really know this picture, know the structures, what they do, and kind of how they're represented in the picture. Again, ribosomes are super important because they are where we build our proteins, and ribosomes are made up of protein itself and a type of RNA called ribosomal RNA. Now again, we find ribosomes basically in two major areas. We either find them attached to the endoplasmic reticulum, and so we call it rough endoplasmic reticulum if it has ribosomes on it. And we also see them floating around in the cytoplasm by themselves. Again, their job, make proteins.